Mansfield College. I send apologies from the principal, who would have liked to have been here. Unfortunately, she's been down with COVID, which of course is ironic given the title of our talk, but we wish her well and a speedy recovery. So it's a great pleasure and honour for me to introduce Sir Peter Horby to you, our speaker for this evening. He is Professor of Emerging Infectious Diseases and Global Health, as well as the Director of the Pandemic Sciences Centre in Oxford. He trained in University College London and went on to specialise in emerging diseases and epidemic-prone infectious diseases. And over the last 20 years, he's made research on many of these things, as I'm sure we'll hear in a moment. He's lived and worked not only in Oxford, but in Southeast Asia for 11 years, working initially for the World Health Organization in Vietnam, before establishing a research centre at the National Hospital for Tropical Diseases in Hanoi. And he was awarded the People's Health Medal by the government of Vietnam in 2016. So he's been back here in the UK since 2014, setting up the epidemic research here in Oxford. He's also Executive Director of the International Severe Acute Respiratory and Emerging Infections Consortium, which is a consortium of 53 international, national and local research networks spanning 134 countries. He leads of course, particularly interesting for this evening, he leads the recovery trial of treatments for COVID-19, which has randomized over 42,000 participants and evaluated 13 drugs. So he's made an enormous impact in lots of different areas, and I'm really looking forward to hearing all about it. So Peter, thank you very much indeed, and thank you for your talk.
Great. Thank you very much, Stephen, and thank you very much, everyone, for joining me on a sunny Friday evening when there are other tempting things to do. So it's great to be here, and thank you for the invite to talk. Thank you. So I'm going to talk a bit about COVID-19 therapeutics and what we've achieved in the last 2 years, remembering that this is a completely novel virus for which we had no vaccines, no treatments, no diagnostics initially, and what we've learned in the last 2 years. I've been working on emerging infections for 20 years, and we've really seen, I think, a sea change in how things have been done. And what we need to do is to make sure that we don't go back to business as normal, that we actually take that level of innovation and catalytic thinking and urgency and apply it every day, and it will make an enormous difference. So I'm going to start by taking you back in time for a call for global action as an outbreak spreads. Taking you back a bit further than you might have thought, this is back to 2009 when the swine flu epidemic first emerged in South America. You can see the great quality of the red tops in terms of the wonderful picture of a man looking like a pig, because it was swine flu. And so we were involved in that. And you've got to remember, this is pandemic flu, which is something that we can expect. It's actually top of the UK's risk register. So there's actually a national risk register of what are the existential threats in this country, from nuclear war to natural disasters, and pandemic flu is number one. So we knew this was coming. How did we do in terms of clinical research, which is my area of work? Well, this is what happened. This is a graph showing the number of patients that were anticipated to be enrolled in clinical trials. So by law, you have to register your clinical trial in one of a number of databases. It's a legal requirement. And so you can look at those registered trials and you could see what was the anticipated number of patients that were going to be enrolled in clinical trials. So when the swine flu pandemic started, there was an ambition to enroll about 4,000 patients into clinical trials to get new treatments for influenza. Now, this might surprise some people, but there are no licensed treatments for severe influenza. The only treatments that are licensed are for outpatients for mild disease. There are no licensed treatments for severe influenza. So there is a real knowledge gap there, and there was a real opportunity to change that back in 2009. But what we saw was just over 500 patients were enrolled globally over that time period. Almost none of the results were published, and none of it was actionable. So more than 10 years later, more than a decade later, we still don't have any licensed drugs for severe influenza. So we missed a massive opportunity there to improve care for a disease that affects us every day and is the most likely next pandemic threat. So it was a real failure. Everyone will be familiar with epidemic curves now. So it really was an epidemic crisis, really, where we had a lot of ideas, a few protocols, but really no patients enrolled and no evidence generated. And what we've been trying to do since then is to shift that curve, to really try and make a difference. When you have an epidemic, it's the only opportunity to advance care for patients with epidemic infections, because it's the only time you can evaluate those drugs. So you have to have a system that means that you can actually operate and do trials under the circumstances of a major epidemic or a major pandemic. And so coming forward to another time when the world must take action, this time it is COVID-19. It's snake flu was what they were initially calling it. I can't even remember why it was called snake flu. So the question is, did we improve our quality of research, even if the quality of the newspapers is still as bad, if not worse? So it was an unprecedented clinical challenge. At the time, if you think back, we were looking at what was happening in Wuhan, China. I had some colleagues there. We were seeing healthcare workers infected. The first picture is Dr. Wen Liang, who was an ophthalmologist, who was one of the first people to whistleblow about the state of the outbreak in Wuhan, for which he was cautioned by the police. Then he succumbed to COVID-19 very tragically as a young doctor. Huge stress on the healthcare surface. In that picture there, the chap in white is a colleague of mine, Bing Cao, who we did some research with. He was sent to Wuhan from Beijing to care for patients, and the patient is actually the hospital director who was sick with COVID. And they published the first clinical description of COVID-19 whilst treating patients and whilst having COVID themselves. So we knew we'd have an overstretched health service. We knew that there would be huge time pressures on staff, and we knew there would be a large number of unwell, anxious, and often elderly patients. And a lot of therapeutic uncertainty. At the time, we didn't know how to treat this at all. We had no therapeutics for coronaviruses. There were many opinions, no reliable data, but a lot of people believing it was reliable. Here is some, one of the earliest observational descriptive studies about the use of hydroxychloroquine, which started this roller coaster and snowballing effect to the extent that you've got the President of the United States saying, I think it works, based on zero reliable evidence. And we're still suffering the after effects of this poorly conducted research being believed by people who really should know better. So this was our early COVID trials experience. This is the epidemic curve in Wuhan. So there's time along the bottom and numbers of cases. And so very, very early on, that chap in the middle photograph, Professor Bin Cao, he rang me up on the 2nd of January, and we decided to try and start a trial in Wuhan because we'd been working together for a number of years. And what we did was we managed to actually start the first trial within 20 days. So even within January, we managed to start a randomized controlled trial in Wuhan, because we knew that if you're going to enroll patients, which you have to do in a clinical trial to improve care, you've got to do it quickly, because the epidemic curves, they're exponential, and so you have to really move quickly. So we started the first trial in 20 days. We then started, the green block here is the first placebo-controlled randomized trial. So we managed to get placebo into China and start the placebo-controlled trials. We had 2 randomized controlled trials in Wuhan in January and February. These were the first 2 trials that were ever started. But what happened was this. So that's what happened to the epidemic curve in Wuhan. So almost at the peak, we opened to enroll for the placebo-controlled trial. And the very next day, the case numbers declined massively. And so we had missed the peak. And we had 2 trials with 200 patients in each trial, which was not enough to give us any reliable evidence. And then the crisis spread. So just as we were seeing that we were not getting sufficient numbers in Wuhan, the outbreak was spreading to Europe, and we were starting to see the very large numbers of cases in northern Italy. And that's when we decided to do the recovery trial. So we took the principles from Wuhan and we decided to move it to the UK. And there were some guiding principles. We knew we had to be quick. We'd seen this before in previous epidemics, that the exponential growth and it moves through populations very quickly. You've got to be very quick to get the upswing. We knew it had to be big. There are a lot of people saying, oh, you know, hydroxychloroquine or ivermectin, it's a miracle cure. We're seeing 50-80% reductions in death. You know, you just don't see that with infectious diseases. There are not miracle drugs like that. You know, antibiotics, you know, I guess, you know, are an exception as a class of drugs. But with viral infections, there really aren't very powerful drugs. And so with an acute viral pneumonia, you're unlikely to find something that's very powerful. But in a pandemic, you know, if you can shift the curve slightly, that has a huge effect. So a moderate benefit in a very large number of people affected will be very beneficial. And we estimated, so we were estimating for a 20% reduction in fatality, which is a very important difference. But many trials were looking at, well, we're going to get 50-80% reduction, which was not realistic. So with 20% case fatality in hospitalized patients, looking for a 20% reduction, you need 2,000 patients per arm. So you need a big study. And it must be simple. Having seen what was happening in the healthcare system in Wuhan and in Italy, we knew that this was going to be an extremely pressured environment. And so whatever we did, it's got to be very simple so that the frontline clinicians, nurses, pharmacists, research nurses and staff could do that under extreme pressure. And so we designed the recovery trial, and what we designed was a trial that was very practical. It doesn't have to be complicated, and this is one of the key lessons. We had a very simple eligibility criteria. If you were hospitalized with COVID, you could be in the trial. Didn't matter how old you were, didn't matter if you were pregnant or not, you could be in the trial. We used a very unbiased outcome, mortality, very simple, very easy to measure. It's not something that's going to be a biased assessment. You know, if somebody's dead or not, it's very easy to tell. And obviously then we randomise. So we have a bunch of treatments. Everyone can come in and we randomise, and then we look at the end of how many people have died in each arm. And the follow-up was just 1 page long, so very, very short. And it takes on average about 8 minutes to randomise a patient to the trial. So the IT system collects this data. So when you first open the IT system to enrol a patient, to the time at which you get the answer, which flashes up about which drug to give the patient, the average is about 8 minutes. So it really is an incredibly simple trial. And we could get away with that in the UK because we could use data linkage. Now, as a first-hand user of healthcare data linkage, it's incredibly powerful and incredibly useful. And there have been controversies about using people's healthcare data for research purposes. But I'm a huge advocate of it. It's all anonymized, so that, you know, we have very strict rules about who can access the data, how it's accessed, what linkages we use. We use depersonalized identifiers. But what it means is that we can get unbiased estimates of death from mortality statistics, of who ended up in intensive care from hospital episodes data, who ended up having to have renal replacement therapy, and we have permission to do linkage for up to 10 years. So we can look at things like long COVID outcomes, cancer outcomes, all of these things we can do from routine data sets. And the NHS and the data linkage now within the NHS and NHS Digital is a real jewel in the crown in the UK. And that's one of the reasons that we've really led the world in terms of finding new treatments for COVID. It's because of this system. And I think if anybody asks me what my opinion is about saying ticking yes in boxes about your data being available, I can promise you there are very stringent rules about how it's used, and it's extremely useful, and it saves lives. So this was the first few weeks in the UK. Again, time along the bottom, numbers of cases, and this is where are we? This is sort of early March, just here. And so we got the funding on the 5th of March. We got funding approval from the MRC to do the recovery trial. We drafted the protocol in 2 days. So on the 7th of March, very simple protocol, but actually there's an art in simplicity. Most of the protocols that you see are very complicated, so we had to really strip it right back. We went to speak to Chris Whitty, the CMO, on the 10th, of March and told him what we wanted to do, and he just basically gave the green light. We had a 10-minute meeting, explained the trial. He said, go ahead, you've got my full support. And so we enrolled the first patient in the Radcliffe just 10 days later, just as we got the upswing of the first wave in the UK. So very rapid initiation. And so this was Bin Cao enrolling the first patient in Wuhan in January. Then a couple of months later, this is my colleague who sort of runs the day-to-day operations of the trial, Richard Haynes, enrolling the first patient at the Radcliffe. So we managed to rapidly switch direction from a trial in Wuhan to a trial in the NHS in the UK. And so this is, again, this was the upswing here, and you saw the first part of this graph in the last slide. And this is the first wave in the UK. You can ignore the sawtooth effect. That's just a lack of weekend reporting of data, so that's an artifact. And so within 2 weeks, we'd enrolled 1,000 patients. Within a month, 5,000 patients, and within 2 months, 10,000 patients. So that critical timing made all the difference. We worked out that if we'd delayed the start by 2 weeks, because we were doing more complex things, which some of the regulators wanted us to do, we would have delayed our primary, our best result, the dexamethasone result, by about 3 months, 3 to 4 months. So it would have made a huge difference. So any delay at the start is magnified in terms of when you get a result. So COVID can affect everyone, so it's open to everyone. It's open to all ages. Our youngest participant is an infant, and our oldest participant is 109. So anyone's eligible, and we include pregnant women as well. And this shows you the recruitment, where we absolutely matched the epidemic curves in the UK. And at our peak in the first wave, we were enrolling 400 patients per day, which is bigger than most trials of COVID that have been done in the world. And in the early part of 2021, we were actually enrolling 500 patients a day. So really huge numbers. And that's because it was operating across the UK, and the green circles are hospitals in the UK that are recruiting into recovery, and the size of the green circles is the number of patients per site. And our biggest recruiter is Leicester with more than 1,000 patients. And you can see up here, this is from yesterday. So total number of patients recruited is almost 48,000 patients. When we initially put in for the trial, we said up to 20,000, so we've well exceeded what we were expecting. And we've now opened internationally. We're in 198 sites, but across many other countries as well. And so what are the revelations? I think we've tested a lot of drugs now. So these ones at the end in sort of the pinky colour are the ones that are still enrolling patients now. And so the others are closed. And we saw, perhaps not surprisingly, things like hydroxychloroquine didn't work, and some other drugs that we might hope have worked, like aspirin, that are cheap and cheerful and help with the clotting problems, also don't work. But surprisingly, what's worked really well is the anti-inflammatory drugs. And that, I think, for me, that's the biggest revelation. This is an infection, and the mantra has been, don't give immunosuppressants like steroids to patients with infections. You'll make it worse because you'll suppress their own immune response, and you'll make it worse. And nearly all of the guidance that was written on treatment of COVID-19 in March, April of 2020 specifically said, don't give steroids. It didn't just say, it wasn't agnostic, it said, don't give steroids, they're contraindicated. And the first result we got was dexamethasone, which gave you 20 to 30% reduction in mortality. So we moved on to other anti-inflammatories. Tocilizumab is a more targeted anti-inflammatory, as is baricitinib, and they both work as well. And what's been amazing is they work in combination. So if you give tocilizumab on top of dexamethasone, you get an additional mortality benefit. And then if you give baricitinib on top of tocilizumab and dexamethasone, that's triple immunosuppressive therapy, you get an even bigger mortality benefit in the severe patients. So that's been the real eye-opener for me, and I think has opened a whole new field really in viral infections, is that actually immunomodulation is a key part of how we can respond to these infections. And we also showed that artificial antibodies, the monoclonal antibodies, are effective, but only in patients who don't have their own antibodies yet. We saw a very striking difference between those two groups. So for those of you who are not sort of medical, we're going to give a little sort of tutorial on forest plots so you can understand the next slide. So this is a way of displaying data from a trial. So along the bottom is your scale for your statistics, so your risk, where 1 is no different and 2 is your risk is doubled and 0.5 is your risk is halved. And so this vertical line here is the line of no difference. And then what we do is we plot our trial results on this. And so the square is the primary, the sort of the median result of the study. It's here saying, you know, whatever your risk factor is, it's giving the drug reduces it by, you know, just over half. And then depending on how big your sample size is, you have smaller or bigger confidence. So if you have a very large number of patients, then your result is quite precise. And if you have very few patients, then it's very wide. It's a very imprecise result. So here you would say, you know, it looks like it might reduce mortality because the square is on this side, but not with much confidence because it crosses 1 of no difference. And this is the results from recovery. It's a bit small to see, but you can see on this side means the drug is improving mortality, and on this side it means it's making mortality worse because usual care is better. And so you can see down the middle there's many drugs that really don't do much at all, particularly hydroxychloroquine, which if anything is on the wrong side of the line. Azithromycin, convalescent plasma, sadly, really didn't have an effect, aspirin, colchicine. But you've got these really nice benefits here. So this is dexamethasone. It doesn't work in patients with mild disease, but it clearly works very well in patients with more severe disease. And here with tocilizumab, the other anti-inflammatory, this is given in addition to dexamethasone, improves survival, clearly on the left and doesn't cross 1. And then artificial antibodies here. If you're seronegative, you haven't got your own antibodies, a very clear survival effect. So the beauty of having a very large sample size is you get these very precise answers which change practice. And because of recovery's been so big and because the answers have been so precise, it has changed practice around the world. So this is this is a pictogram of the WHO treatment guidelines from March, which are updated regularly. Across the top here is different levels of severity. So I've blanked out this first bit here, which is outpatients, because that's not relevant to our trial. And these last 2 bits are inpatients. And you can see the things that are recommended, these 4 drugs here are recommended, you know, largely because of recovery, or partly because of recovery, but mostly because of recovery. And there are things that are specifically not recommended, don't give them. And that's for our results as well. So here you can see 7 results from recovery that have gone straight into WHO guidance. So the revolution, trying to shift that curve. You know, remember this from the start, the 2009 influenza pandemic. No actionable evidence from a pandemic we knew was coming. We really shifted the curve. You know, we anticipated enrolling 20,000 patients. We've now got nearly 48,000 patients enrolled. You might think strangely, we've published on over 80,000 patients. How did we do that? It's because we used a factorial design where you could be in more than one arm at the same time. So you could be randomized to aspirin or not, but also to dexamethasone or not, because they have different mechanisms of action. So you could contribute to both of those comparisons. So by using this clever factorial design, we actually get more sort of bang for our buck. And so we've published data on 80,000 patients, and it's all been actionable. Every single one of our results has resulted in change in practice, either to be taken off the recommended list or to be strongly recommended. But there's also other problems we have. You know, we did this trial in the UK mostly, although it's now going overseas. But there's a real problem with the fact that the clinical trials are focused in high-income countries. This is a 2020 picture showing the distribution of clinical trials around the world pre-COVID, and you can see, you know, there's hardly any research going on in lower-income countries. You know, Central Asia, Africa, South America. There really is a divide in where the clinical trial data is coming from. And this is a real problem because you've got to have data that's applicable to your local population, because your local population is different in age, in comorbidities, and all sorts of other ways. And your healthcare system is different. So, you do need to address this imbalance. And this is another way of showing the distribution of clinical trials for COVID. And this is the high income countries, upper middle income, lower middle income, and low income countries. And the number of countries and the size of the trials. And you can see the vast majority of trials, again, high income countries. As you go down, fewer and fewer, and no trials in low income countries. So it really hasn't changed, unfortunately. And this is something we need to tackle next. I think we've found the methodologies that we can do large-scale trials, but we need to transfer that to different healthcare settings in lower income countries. We're trying to do that, so we've now opened Recovery International because of its success. We're now open in Ghana, India, Indonesia, Nepal, South Africa and Vietnam. Different challenges there. We don't have the health data linkage, so we have to use other mechanisms for getting to reliable estimates of death in a simple way, etc. But we're really using this now to sort of try and roll out this idea of a platform trial, a trial that's very simple. You can put drugs, lots of drugs through it, up to 13 drugs so far, keep it running, but do it in low-income countries as well. We've had some very nice quotes about introducing the ethics committees to new ways of doing trials, because platform trials are a fairly new way of doing things, particularly for infections. And then there's the efficiency crisis. The reason that trials aren't done in low-income countries is also the cost. It's quite astronomical, really. The median cost of a clinical trial is about $19 million. For one trial of one drug, $19 million. And if you have an active control, which means that it's not just it's one drug versus another active drug, it goes up to 50 million. If you have a clinical outcome like death or hospitalization, as opposed to some sort of blood biomarker like what happened to your serum level of a protein, if you have a clinical outcome, median cost is nearly $65 million for one trial. These numbers are jaw-dropping, really. So that's a mean per patient of around $36,000. It's just unaffordable in most settings. And the time to complete trials about 40 months. So huge amount of money and a long time to do clinical research. And yet a lot of that money is wasted. So this was an FDA review of 2,000 registered trials for COVID. And they looked at them and they analysed them and they decided, were they adequately powered, by which we mean, are they big enough to give you a reliable answer? As I said before, if you've got small numbers, you're going to have an imprecise answer, which is not going to change practice, because they say, well, it could be chance. We're not going to start treating everyone with this drug when there's a lot of chance at play. And 95% of the trials registered for COVID are not powered to give you a reliable answer. So you're not only spending huge amounts of money, you're wasting huge amounts of money, because 95% of the trials are not going to give you a reliable answer. So it's almost criminal, the money that's being wasted on these poorly designed trials. And if you look at those sort of numbers versus what we achieved in recovery, the average time for funding to be agreed to a first patient being enrolled for some cancer trials is about 621 days, whereas we managed it in 14 days. Now that required support from government, from the NHS, and the health infrastructure in the UK, but it shows you what can be done. You can really telescope this hugely. 3 years to get a result for many phase 3 clinical trials, the late stage clinical trials. We got our first result in 3 months with dexamethasone, very quick. 2,000 trials with no answers, underpowered. We've already, just for one trial, got 10 answers that have answered questions. And our average cost per patient is $500 as opposed to $36,500. So obviously the pandemic was an unusual situation, and we can't expect everyone to drop all their other research and to put everything into one trial, which is what happened. But there's got to be a middle ground between this extreme situation and this situation where we can do much better, and do that not just for infections, but do it for cancer, for hypertension, for stroke. It shows us that there is a much more efficient way of improving health. So us and others, we're pushing about reimagining randomized trials. They've got more and more complicated, so that they're hugely expensive. They take years and years. You have to employ these contract research organisations which cost hundreds of thousands of dollars to run your trials, and it doesn't have to be like that. It can be much more simple. It can be at the interface between the healthcare staff and the patients, using data that's already routinely collected for free to look at your clinical outcomes. But it needs to be supported by proportionate trial regulations and guidance. You know, the current guidance around clinical trials is very, very complicated, and a lot of it is not critical to quality and can be got rid of and changed. And that way we'll have, you know, a lot more benefits for the patients and the public health and for the public purse. So this just shows you what happened with recovery. So along the bottom here, it's not really ordered, but these are hospitals. Each blue column is a hospital. And the height of the blue bar tells you how many COVID patients they admitted. And the dots tell you what proportion of those patients they enrolled into the trial. And you can see some were doing a fantastic job, enrolling 30% of patients into the trial, of all patients with COVID. And overall, about 10% of all patients in the UK were enrolled in the trial, which makes sense because there were no treatments and people wanted to be offered a treatment, and healthcare staff wanted to be able to offer something. And the way to do that was to put the patients into a simple trial. Then you had a choice of potential treatments. And now the patients enrolled in the trial now are benefiting from the patients who went earlier because they're getting the treatments that were evaluated earlier. So there's been a lot of positive things said about the recovery trial. But I think the real one for me is this picture, which was sent to me by one of the clinicians, which is an intensive care admissions board. Anyone who's been in a hospital will see this. You know, there'll be a ward with the patient's name here, you know, when they came on the ward and what their diagnosis is, and you know, who's treating them and what their treatment is. This was a COVID ward. Obviously, everybody's got COVID, but they've added a column for which trial the patient was in, which is completely novel. You know, this trial's seen as something separate, but this just shows you how integrated into care that the trials were, that you're putting on the board for every patient. Which trial are they in? They need to be in a trial because that way, you know, we'll learn and we'll improve care. And so just to finish, to say that, you know, COVID may be something we all wish to forget, but there are plenty more infections out there. And this is a pictogram of somebody who tried to add up all the deaths in the 20th century and what caused them. And you can see this huge circle here for infectious diseases. And that's war there in a century where there were some pretty big wars. Infectious diseases, you know, cause a huge impact in terms of deaths. And we've seen with pandemics, not only that, they have a huge impact on socioeconomic welfare, and beyond the direct impacts, they have many, many indirect health impacts. So we really need to invest in making sure all the lessons we've learned from the COVID pandemic in terms of research around vaccines and treatments and public health interventions are not lost. They have to be embedded into routine practice so that when this happens again, we're ready to turn on the tap straight away. And so just a plug, so we're setting up the Pandemic Sciences Institute. This is going to be a new building on the Old Road campus where we're going to bring together multidisciplinary groups. We've got, you know, right from basic science and structural biology through to clinical trials, vaccinology, but also social sciences, behavioural sciences, ethics and history will all be under one roof, looking at how we can tackle not just pandemic threats, but epidemic infectious diseases in a way that has utility day to day, so that we can help people, you know, in the absence of an epidemic or pandemic, but we have the systems in place so that when these things happen, we can respond very quickly and make a difference. So just to acknowledge, obviously the recovery trial has been run by the NHS, and it's huge thanks to all of the healthcare staff around the country who've made it possible, but also to all the patients and their relatives who've agreed for them to be in a trial when they've been under a lot of stress. So thank you very much for your attention.
Please do have a seat. Well, thank you very much indeed. That was absolutely fascinating and really inspiring. I'm sure I speak for everyone when I say that. I'm going to open this up to more general discussion, but I just have a few questions myself, actually, just to begin with. First of all, I mean, you mentioned with the dexamethasone that there were good reasons for believing beforehand that it wouldn't work. because it was against the sort of current understanding. Do we now understand why drugs like dexamethasone work?
Well, it was kind of a brave move, really. We had thought that, in severe, particularly novel viral pneumonias or viral infections, there's a lot of inflammation, and I'd seen that in patients who had avian influenza, so H5N1, these very severe influenzas. And the early picture was that this was a bit similar, a very inflammatory pneumonia. So we believed that was part of the pathological process. But we got a lot of criticism. We actually got letters. I got a letter sent to me from 10 or more eminent professors in London, copied to the chief medical officer and the chief scientific advisor, saying that we were unethical putting dexamethasone into the trial and it should be stopped. So it was a nervous time when we were putting patients into it. So when we saw the results, I was greatly relieved. But I think it's about the timing, and the dose is really critical. I think with any drugs and infectious diseases, you're at different stages. And in the early stage, your immune system is trying to fight the virus, and at that stage, antivirals are really important. But by the time you get to hospital, you've got the viral replication has driven a lot of pathological response and inflammation, and at that stage, you need the antivirals plus the immunomodulators. And what we saw was that in patients in very early disease who didn't need oxygen, there was no benefit, and it was consistent with harm. So one thing that does worry me is we're now seeing over-liberal use of steroids, and I hear that in some countries like India and other places, steroids have been given to people very early, even in the outpatient setting, and I think that's a bad thing. So we have to temper people's enthusiasm.
Right. Thank you. And you mentioned that with pandemic flu, which is number one on the risk register, we still have no treatment for severe disease. So if we were to have a pandemic flu outbreak, do you think the lessons learnt from what's happened with COVID would allow something to be developed, or is it a...
You would hope so, right? But so we've put flu into recovery. So we now have an arm where we could enrol flu patients. But to be honest, that was real hard work. It took a lot of convincing to let people do that. One of the arguments was, but it's called the recovery trial because it's got COVID in the title, so you can't put flu in it. It's just such an absurd argument. But that was from a regulatory body. This is just stupid, And then we're having trouble now financing flu in recovery. So what we are seeing is a really rapid return to business as usual, which really worries me a lot.
Yeah. So I'm going to open it up for questions. Catherine, would you like to wait for the microphone? Thank you.
Thank you. First of all, I'm in awe of the work that you did and are doing throughout the pandemic, including ignoring silly letters from people who should know better, saying don't even try dexamethasone. But to my question, I think I heard you say towards the beginning of the talk that there are no magic antivirals, there's nothing that's remotely an analogue of antibiotics. Is it possible to explain to the non-specialist if that cannot be at all, or whether it's a case of more work, more research, more time?
Yeah, I mean, there are, I think for sort of chronic viral infections, there are good antivirals. We've seen the remarkable success of antivirals for HIV and some of the hepatitis viruses. But for these, acute, short-lived, quite severe viral infections, then the replication is so quick and it's intracellular, unlike bacteria's. So it's much more harder to target them. They have a very high replication rate. They're simple organisms, so they evolve very quickly, so you get resistance very quickly. So they are challenging. But they do have conserved parts to them. So some of the internal replication machinery is very conserved. And so if you can tackle that, you can probably have antivirals that will have an effect if you give them early enough, and they're unlikely to be susceptible to resistance. But I think they are harder, and what we haven't seen is really the investment in them either, because they're not really seen as a big market. The drug companies are spending money on better lipid-lowering drugs and better antihypertensives and making marginal gains in those areas rather than viral infections.
That's a pity. I wonder if I could also ask, would it make any difference if you knew what the origin of this virus was, whether it sort of naturally jumped from whatever it's supposed to have jumped to, or whether it was an unfortunate lab experiment or accident?
I don't think it would have made a lot of difference. I think what we need to do, I mean, we knew about coronaviruses in various species and bats, etc. But there hadn't been, and we knew about SARS-1, and that was, we just dodged a bullet there. It's almost exactly, the inception stage is almost exactly the same. It came from a wet market, probably. It started to amplify in a hospital. We started to see healthcare workers die. It spread internationally, but it just wasn't quite transmissible enough. But it was a whisker away from being, a pandemic. But in those 10 years, 20 years, there wasn't very much investment, really. People rapidly forgot about it. And so, I think we could have been better prepared. There were a few people, like Sarah Gilbert, had the MERS vaccine, which is close to SARS, and there was a bit of data about antivirals for SARS-1, but not very much. And that's where I know times are tight and money's short, etc. But we really need to find the money to make sure that we look at all of the pandemic threats and invest properly, so that we have a good understanding of how they work, that we have the potential antivirals to test and vaccines to test.
Yeah, there's a question at the back.
Thank you for a fantastic talk. You mentioned that patients could be randomized to receive or not multiple drugs at the same time because of independent mechanisms. Mm-hmm. At this point of uncertainty over what was going to work and what wasn't, was there also uncertainty over drugs possibly interacting with each other? And did that sort of limit some of the combinations you were thinking about?
A bit. We moved towards the factorial design, which is so, you have different designs where you could have drugs in parallel, so you could be randomized to drug A or drug B or drug C or drug D, and we started like that. So when we started, you get usual care or hydroxychloroquine or azithromycin or oral lopinavir, ritonavir, an HIV drug. But then when we came to put in, say, aspirin, we thought, well, really that is a completely different mechanism of action. These are supposed to be antivirals. This is an anti-clot drug, and there shouldn't be any interaction. So then you could be randomized to both of those things simultaneously. But we do look for interactions. So what we do is when we look at the result, we look at whether the effect of drug A is different, whether you've got drug B or not. And so, again, the beauty of recovery is the size of it means we've got enough patients to do that. With a small trial, you can't look at those kind of interactions, but we could. And so far, we perhaps have seen an interaction. What's interesting is the second anti-inflammatory drug, tocilizumab, that we showed worked. Most patients were on dexamethasone, but there were some who weren't, and that didn't look like it had an effect. So it's actually been licensed only on a background of dexamethasone.
Right, other questions? Yes, that one there.
Thank you for the presentation. The question I have is that during the trials, a lot of vaccines and drugs that were tested, they were tested in a very short duration compared to generally how the trials are done over, like, for example, you showed 3 years, so you could shorten the period. So does it have any effect on the long-term potential effects that the vaccines or drugs may have? And whether going in a similar fashion, if in future, like, the trials are done on a shorter duration, it may have some potential long-term effects?
Yeah, thanks. Well, it shouldn't do, in the fact that it's the same number of patients. They're just the time that they're recruited over is compressed, so it should have no effect on to the long-term effects. But I think it is important that we have long-term follow-up of patients, and one of the things we're very interested to look at is whether immunomodulation of patients who are hospitalised with COVID affects their subsequent risk of getting long COVID or not. The only caveat to that is that with infectious diseases, the organism evolves. And so if you test your drug at this time point and the pathogen changes, is the result here still relevant here? And we've definitely seen that with Omicron, with the variants. So we tested a monoclonal an artificial antibody against Alpha and Delta, and it worked. And it was registered for use, but then we had Omicron, which the genetic data suggests is resistant to this drug. So there is an issue about the timing when it comes to drugs that evolve.
There's a question at the front here. Thank you.
Good exercise. Hello. I wanted to raise the issue of the waste that you showed on one of the slides in a lot of the studies. We know it's there, but you showed a review study that really showed this. I chair a working group looking at AI applied to health interventions, and it's the I2WHO focus group for AI for health. And one of the studies we looked at was the use of image data in COVID-19, and it was like there were 2,000, the review found 2,000 studies in the period of COVID, narrowed it down through a set of criteria, you know where I'm going, several hundred passed the criteria, 60 made the study, and there's this wonderful line that says, at the end of all of this, none of this was clinically useful.
Yep.
And you think, what's going on there? This stuff gets published, it gets funded, and we're trying to think there must be some way of saying that at some point you have to jump through some hoops. There has to be some criteria. Just to get published, there should be some criteria, or these things should be flagged. And when these trials are registered, is there not some way you can sort of have some, or is it more too complicated, or what? Because it's not just a waste of money, it's a waste of opportunity costs. Those researchers could be doing something useful and having the impact that you guys have been having.
Yeah, and it's unethical, because all the patients are, you're wasting patients' time and effort and hope, on studies that are not going to tell you anything. And it's just an awful story, isn't it? That you've got all of this money and all of this time and all of this goodwill spent on useless studies that don't tell you anything. And some people have been saying, because it's I guess that problem's true across many areas, but it's been most highlighted for the clinical trials, that there should be coordination. But I think actually there's a lot of culpability in the ethics committees that pass the trials and the scientific review of these trials. Like who reviewed 1,950 trials that were underpowered and said that's fine, go ahead, and who funded them? The funders. And I think there's a lot of culpability there, that instead of the funders saying to us, oh, you need to coordinate better, saying to the funders, no, you need to stop funding rubbish research and put it into large-scale stuff. If all of that, I bet if all of those images had been pooled, it would have had a clinically...
That would be the next stage, if you think of some of our colleagues who pool underpowered studies and make them more powered. I don't know whether that's a part of the solution.
Yeah, so I think, there needs to be initiatives to get people to filter out the stuff and collaborate more.
Thank you.
Please go ahead.
Thank you very much for your talk. I have a question. It's more rather about the people who go online and spread some conspiracies and some lies. And do you think that there should be legal action taken against those people? And how do you think that we can improve the situation? Because in the UK it was fine, because we have a very high vaccination rate. But in other countries like Germany or the US, it was not so good.
Yeah, it's a very difficult area, isn't it? I've had my fair share of, people have, said I should either, go to Stockholm for a Nobel Prize or go to the International Criminal Court to be tried for genocide, and they're both ridiculous ideas. But there's been lots of online abuse, saying we're murderers, and murderers because, what we tested, and murderers because we didn't test something, or being told that we faked the results of hydroxychloroquine because we were being paid by Bill Gates. And there was a guy camped out in our building for 2 weeks with a thing saying that we faked the results and Bill Gates is paying us. And okay, we do have a grant from the Bill and Melinda Gates Foundation, but that's a bit different. And it wasn't even for this study. But I don't know what to do about it, because I have tried answering some of these people on social media, and it doesn't help. It just amplifies their message. And now every time I tweet, I can almost know who's going to respond, because there's always sort of a certain group of people who respond saying, yes, but you should have tried our drug, and things like that. So I don't know, and I don't know whether, this is a question for other groups really, and that's one of the reasons we're getting the sort of social behavioural scientists and things into the Pandemic Sciences Institute, because it is really important, because there's no point having a drug or a vaccine that people won't take, or politicians that are spouting rubbish because they're not advised properly. So you need to bring all that along as well. It's not just about the science, it's about the science communication too.
One thing that came out, just sort of be a slightly more positive slant, something that came out very strongly from your talk was how important data linkage was within the UK system, and something we've clearly got right, that the various bits of the health service are talking to each other so you can harvest that data, and that was a problem in lots of other countries where that data linkage doesn't exist. Do you get a sense that there are other countries who are starting to say, wow, the way the Brits do this could be useful, we need to reorganise our health service so that we can have this data linked better?
Yeah, so we've been asked to talk in the US and Canada and Singapore and Japan, and about how we manage to run it. It's incredibly efficient compared to traditional ways of doing trials. For lots of reasons. It helps you with managing your healthcare system, and it helps you also if you want to do research efficiently. And it would apply to anything, because those data sets tell you what people died of anything, so you can use it for cancers and all sorts of things. So I strongly encourage that people get that sort of data access, but it's an increasingly difficult area, I think, because of concerns about privacy, etc. So you do have to have a trusted provider, and I think we are a good example of how of the good that can be done from data linkage. We also need to bring the regulators with us, because we've been audited quite a lot by regulators who want to approve some of these drugs, and they've said to us, how do you know the patient's dead? And we said, well, because we have a death certificate. And they said, yeah, but this form filled in by someone on the ward in the hospital doesn't say the patient's dead. Saying, no, I know, but they are dead because there's a death certificate. And they said, well, how do you know they're dead? Because it's signed by a doctor. And do you want us to exhume the body? They're definitely dead. But they wanted to go back to to a piece of paper, and so they're struggling a bit with the idea that data linkage is a reliable thing. And we've just today submitted a response to one of the regulators showing them that actually the data linkage was more accurate than the forms that are filled in on the wards by healthcare staff, and so we should believe in it.
Right. Is there an understanding? I guess health services are very large bureaucracies and quite difficult to move and change, and that must be a challenge. Well, it's a challenge in every country. How did we get to the state where data linkage is good in the UK? What's that a product of historically?
Yeah, it's not my area of expertise, because I'm actually tropical medicine. This is the first trial I've done in the UK, but there's been a big investment in electronic health records, and an organisation called Health Data Research UK, HDR UK, and NHS Digital. So there's been a big investment that somebody saw the value in digital healthcare, not just for running the healthcare system, but for other purposes as well. And so I think it's been hugely beneficial. Yeah.
Right. Any other questions? Yes.
Yes, so thank you for that talk. It's very interesting. As you mentioned during your talk, that timing and scalability is everything when it comes to clinical trials, and you had support from the government and the regulators in accelerating that. And normally a clinical trial takes a lot longer. What lessons do you feel that have been learned for other clinical trials moving forward as a result of this clinical trial for COVID-19?
Thanks. I think one lesson is that the free market, it doesn't really work when it comes to sort of drug development, to be honest. So the drug companies investing in drugs that are going to be for chronic diseases in rich populations, so they can treat people for 20 years with a drug that costs thousands of pounds. So, so they're going for, as I say, cholesterol lowering and hypertension and things like this. The idea of investing in a drug that's, for acute respiratory infection that, may or may not occur isn't in their business model, so it doesn't get done. Whereas the government, I think, should be strategic about which trials they support. They did that for our trial, and so there was a letter from the chief medical officers of all the devolved nations and the NHS chief executive saying, please support this limited number of trials. And now, if you imagine if they did that for dementia, or heart disease, there's enough dementia patients to have a massive platform trial for dementia. Why isn't there one? It should be a national priority to do that, and it could be done. And it could be done at a fraction of the cost of one of the dementia trials done by industry. But you need a sort of a less laissez-faire attitude towards it. It has to be a directed research program from government.
Thank you.
And I mean, one thing you also mentioned was the fact that we're starting to forget that there was a pandemic, and so therefore the urgency that maybe we all felt a year ago, which has led to the funding of a whole range of different things, is that urgency is reducing. So, yeah, what do you think needs to be done either in society or in government to prepare for the next?
Yeah, I guess I don't see the urgency there in the political arena at all. There has been some documentation about this. We were presidency of the G7, and some documents were produced around the 100 days mission, where 100 days, a mission that within 100 days of a new pathogen, we should have a diagnostic, a vaccine, a drug ready. And some documents were produced, but that's not been followed up with funding. It seems to me there was a lot of empty rhetoric there. And so I don't see it coming from government, actually. And I think it needs to be more of a grassroots kind of activity where the healthcare community, the research community, and the general public are saying, look, this was devastating. We can't accept this happening again. And what are you doing about it? And you've said that we should do this in 100 days. So what are you doing to make it happen? So a lot of advocacy, I think, is needed, but it's going to be a hard push because there's so many competing priorities at the moment for a strapped budget.
Any other questions? Yes, one there. The final question. If you could just wait for the microphone, sorry.
Do you think the regulatory system can cope with a pandemic type emergency?
It was a real mixed bag. The regulators were fantastic. So these are the drug regulators that approve trials and approve drugs and things. They were fantastic at some points. They approved the trial very quickly. In a few days, and they approved of all our amendments and all our new drugs very, very quickly. Other regulators weren't as good. The US Food and Drug Administration approved hydroxychloroquine for emergency use based on absolutely no data. So there was good and bad political pressure. So, there was a political pressure, I think, on the regulators in the UK to facilitate some good level trials and some bad political pressure in the US. But they're still going back to a system that after the pandemic, a regulatory framework, a legal regulatory framework that to me doesn't seem fit for purpose. It's got more and more complicated. And we have the EU clinical trials regulations and we have good clinical practice regulations. And they are, I think, absurdly restrictive and complicated, and they need to be completely revised, in my view. I think there are good people in the regulatory system, but they are bound by, the laws that they're supposed to be enforcing. And so I think those laws and regulations need to be reviewed.
Okay, well, thank you very much indeed. Before we thank Sir Peter, can I remind you that the next Mansfield Public Talk is actually not on Friday next week. Unusually, it's on a Wednesday. So we have David Goodman of Boston College, who's going to be talking about the streaming south, the joining effects of technology and liberalism. And next Friday, there is another event as well, Friday the 20th of March at 5:30 p.m. There is Dreams of Peace and Freedom, a Justice for Ukraine concert, a performance in support of the international petition demanding a tribunal to enact justice for victims of Russian aggression in Ukraine. So 2 events for next week. But now will you join me in thanking Sir Peter for an absolutely wonderful and inspiring lecture. Thank you very much.